Today we're going to go over some practice questions in our eukaryotic cell unit. So as always, feel free to pause in between and give a justified answer to why each one is correct or incorrect. Start off with number 1. Which of the following structures is responsible for modifying and packaging proteins for secretion? The rough ER, the Golgi, the smooth ER, or the nucleolus? This should be a straightforward one, as the answer is the Golgi. Number 2. What type of vesicle coat protein is primarily involved in enterograde transport? Clathrin, COP1, COP2, or dynamin? And the answer here is COP2. You should note that enterograde transport is from the ER to the Golgi, and COP2 is anterograde from ER to the Golgi. COP1 is retrograde, so from the Golgi to the ER. Moving on to number 3, which organelle contains hydrolytic enzymes and maintains an acidic internal pH? This should be a lysosome. Number 4, which cytoskeletal element is involved in the formation of the mitotic spindle during cell division? Is it microtubules, microfilaments, intermediate filaments, or actin? The answer here is microtubules. So recall that intermediate filaments don't have a specific role in cell division aside from just transporting things. And microfilaments are made of actin, and that forms a cleavage furrow, and microtubules is what actually forms the mitotic spindle. Moving on to number 5. What is the primary role of peroxisomes in the cell? A. ATP production via oxidative phosphorylation. B. Detoxification of hydrogen peroxide. C. Protein synthesis. Or D. DNA replication. For this one, I hope you knocked out C and D right away because it's not protein synthesis and it's not DNA replication. And it does not produce ATP via oxidative phosphorylation either. The main role is detoxification of hydrogen peroxide. Number 6. The double membrane and presence of circular DNA are features of which organelle? And this is the mitochondria. Recall the endosymbiont theory says that mitochondria was an ancestral prokaryote that a eukaryote engulfed, and so it would have circular DNA because it was prokaryote, and it also has a double membrane. Number 7. Which of the following does not describe connective tissue cells? A. They account for most cells in muscles, bones, and tendons. B. They secrete substances to form the extracellular matrix. C. In organs they tend to form the stroma. Or D. In organs they tend to provide support for epithelial cells. So we want to see which of these do not describe connective tissues. So let's see which ones do. Let's start with C and D. So C says they tend to form the stroma, and D say they provide support for epithelial cells. And we know that stroma is essentially providing support. So both of these things is a function of connective tissue cells, so it's not C or D. Now B says they secrete substances to form the extracellular matrix. And recall that that is exactly what we talked about. So the answer must be A. And A says that they account for most cells in muscles, bones, and tendons. And they do not account for most cells in muscles. And so this is the wrong answer. So A is essentially the correct answer. Moving on to question 8. Which of the following is not a function of the smooth endoplasmic reticulum? Lipid synthesis, poison detoxification, protein synthesis, or transport of proteins? And the answer here is protein synthesis. So they do synthesize lipids, they do detoxify poisons, and they transfer proteins from the ER to the Golgi. Number 9. Which of the following activities occurs in the Golgi apparatus? A. Synthesis of proteins. B. Modification and distribution of proteins. C. Breakdown of lipids and carbohydrates. Or D. Production of ATP. And this should once again be simple. It is modification and distribution of proteins. Number 10, and choose as many as you want in this one too. So mitochondrial DNA is A. Circular, B. Self-replicating, or C. Single-stranded. And so it is circular, and it is self-replicating, but it is not single-stranded. Number 11, what is the main function of the nucleolus? A. Ribosomal RNA synthesis. B, DNA replication, C, cell division, or D, chromosome assembly. And this is also should be basic. You should note that it is ribosomal RNA synthesis. The nucleolus is a little area inside of the nucleus, and this is only for ribosomal RNA synthesis. Or I guess it's from other things too, but primarily for ribosomal RNA synthesis. Number 12. Which of the following organelles is surrounded by a single membrane? A, lysosomes, B, mitochondria, C, nuclei, or D, ribosomes. And so ribosomes are not surrounded by any membranes. Nuclei and mitochondria are both surrounded by two membranes, and lysozyme is the one that is surrounded by one membrane. So the correct answer is a lysozyme. Number 13. Which of the following is not a difference that would allow one to distinguish a prokaryotic and a eukaryotic cell? A, ribosomal subunit weight. B, presence of a nucleus. C, presence of a membrane on the outside surface of the cell. Or D, presence of a membrane-bound organelles. And let's go through this question step by step. So it says, which would not allow us to distinguish between prokaryotics and eukaryotes? So A says ribosomal subunit weight, and this would allow us to distinguish because they have different subunit weights. B says presence of a nucleus, and this is also a distinguishing factor as prokaryotes do not have a nucleus. D, presence of membrane-bound organelles. Well, this is also a distinguishing factor as eukaryotes have membrane-bound organelles and prokaryotes don't. But C, presence of a membrane on the outside surface of the cell. Well, both of the cells have this, so you cannot distinguish based on that. Number 14. Which of the following does not contain tubulin? A, cilia, B, flagella, C, microfilaments, or D, centrioles. Now, tubulin is used in microtubules, and we know that cilia and flagella have microtubules, so it's not that. Centrioles are also made up of microtubules, so it's not that either. Microfilaments are made up of actin, so the answer here is microfilaments. Number 14. If an enzyme that degrades mRNA is injected into the cytoplasm of a cell and all translation ceases, is the cell prokaryotic or eukaryotic? Now this is a little bit of a trick question because translation happens in the cytoplasm for both eukaryotes and prokaryotes, so it could be either. Number 15. When an enzyme that degrades DNA, a DNase, is incubated with intact DNA isolated from an organism, the DNA is degraded. But when that same DNase is injected into the cytoplasm of a cell from the same organism, no effect on the genome is observed. Which is the best explanation for this? A, the cell is a prokaryote, therefore the genome is inaccessible to cytoplasmic enzymes. B, the cell is a prokaryote, therefore the circular genome is resistant to DNase. C, the cell is a eukaryote, therefore the genome is inaccessible to cytoplasmic enzymes. Or D, this cell is eukaryote, therefore the linear genome is resistant to DNase. And the best explanation for here is probably C, that it's a eukaryote and the genome is inaccessible to cytoplasmic enzymes. This is because the DNA is probably in the nucleus, and cytoplasmic enzymes cannot go into the nucleus without a localization sequence, which they do not have. And number 16. If a retrovirus inserts its genome into regions of heterochromatin and nowhere else, how is this likely to affect the infection process? And so first you should know that heterochromatin means densely packed chromatin. And so if it's densely packed, that means it's very inaccessible. And so the retroviral genes will not be expressed very frequently, and the virus will tend to remain as a provirus unless a change in the surrounding heterochromatin allows viral genes to be expressed. Number 17. Would you expect the nucleolus to be larger in cells that are actively synthesizing proteins or in quiescent cells? And this should be in synthesizing proteins because nucleolus is for ribosomes and ribosome synthesized proteins. So if a cell is actively synthesizing proteins, you'll be making more ribosomes and your nucleolus would be larger. Number 18. What role would the loops of DNA in the nucleolus play? These loops of DNA, they serve as a template for ribosomal RNA production. Number 19. So we know that the nucleolus is the site of transcription of ribosomal RNA by RNA polymerase 1, and transcription of mRNA and tRNA is performed by other polymerases in other areas of the nucleus. Now, does a similar division of labor exist in the prokaryotic cell? And the answer is no. Bacteria only have a single kind of RNA polymerase, which is responsible for all transcription. Number 20. Is the space between the inner and outer membranes of the nuclear envelope contiguous with the cytoplasm? This is no, it is not. The space between the nuclear membranes is contiguous with the ER lumen, which is isolated from the cytoplasm. Number 21. If a 15 kilodalton protein has a nuclear localization sequence that is then deleted from its gene, will the mutated protein still be found in the nucleus? Yes, it will, because the protein is small enough that it will still pass through the nuclear pores by diffusion even without a nuclear localization sequence. Number 22. Which one of the following proteins would not be found within the nucleus? A protein component of the large ribosomal subunit, a factor required for splicing, a histone, or an amino acid tRNA synthetase. Now we know the protein components of ribosomes are synthesized in the cytoplasm and then imported into the nucleus to be assembled. So it's not a protein component of the large ribosomal subunit. A factor required for splicing, recall that splicing also happens in the nucleus, so it's not B. A histone, well, histones are obviously used in the nucleus, so it must be D, an amino acid tRNA synthetase. And this is correct because their function in the cytoplasm is to attach amino acids to their respective tRNAs. And so this is why they are never needed in the nucleus and will not be found there. But even if you did not know what those were, you would still be able to round that answer out because of the other ones. You note that A, B, and C are all found in the nucleus. Number 24. A researcher injects tiny gold beads into a cell and waits an hour. Then she examines the cell and finds the gold beads in the cytoplasm and in the nucleus. Now when she injects larger gold beads, they are not found in the nucleus. However, when she binds the larger beads to a nuclear localization sequence, they end up in the nucleus. One can conclude from this that A, the nuclear localization sequence is lysine-rich, B, gold beads have an inherent import signal, C, the nuclear localization mechanism is non-specific enough to confer nuclear import on gold beads, or D, nuclear import relies primarily on simple diffusion. Now we know the sequence is basic and it might be lysine-rich, but we cannot conclude that based off of this information. So it's not A, and they don't have an inherent import signal because they didn't go in when they were not bound to the signal sequence. They only went in to the nucleus when they were bound to the signal sequence. And it doesn't rely primarily on simple diffusion because although it did go in to the nucleus when they were smaller sized, when they were bigger sized, they didn't go in and then they went in with the sequence. So the only option that's left is C, which says the nuclear localization mechanism is non-specific enough to confer a nuclear import on gold beads, which is true. Number 25. Why is the inner mitochondrial membrane folded into cristae? And this is because the folding of the membrane increases its surface area, and this allows for increased electron transport and ATP synthesis per mitochondria. Number 26. If a woman has a disease caused by an abnormality in her mitochondrial genome, what are the chances that her children will have the disease, assuming her mate does not have the disease? And all of her children will have the disease because mitochondria is inherited from the mother. So it doesn't matter if the father has a mitochondrial disease or not, it depends on the mother. And if she has it, then the children will get it. Number 27. The mRNA for secreted protein encodes a longer protein than it is actually observed in the cellular exterior. Why? So A, the protein was cleaved by a cytoplasmic protease. B, the mRNA was not spliced properly. C, the gene encoding the protein contained a nonsense mutation. Or D, the signal sequence of the protein was removed in the rough ER. Now the only way a protein can be smaller than would be expected from its mRNA would be if some post-translational modification were to occur. Now choice B and C are pre-translational modifications, which is splicing and nonsense mutation. So this would not account for a size difference between the mRNA and the protein. And A says the protein was cleaved by a cytoplasmic protease. Well, this is also wrong because they're secreted in the rough ER and they're inaccessible to cytoplasmic proteases. So the answer is D. The signal sequence of the protein was removed in the rough ER. And we know that some post-translational modifications happen in the rough ER, and so it was cleaved there. Question 28. If vesicle fusion with the cis Golgi was inhibited, could plasma membrane proteins still reach the cell surface? And they could not. This is because secretory proteins must proceed via a specific path from the ER to the cis Golgi, to the medial, to the trans Golgi, and from there to the cell surface. So if you inhibit cis Golgi fusion, then they will not reach the cell surface. Number 29. Cardigener syndrome is a rare genetic disorder that results in immotile cilia. The immotile cilia cause infertility, bronchostasis, and recurrent sinusitis. The genetic defect causes the deficiency of a protein primarily involved in which of the following structures? A. Microfilament, B. Intermediate filament, C. Microtubule, or D. Plasma membrane. And this should be microtubule because recall that cilia has microtubules in it, so that's what would be affected. Number 30. Different proteins have been found to be involved in vesicular trafficking. Specific proteins are responsible for specific pathways. For instance, COP1 is responsible for retrograde transmission of vesicles, while COP2 is involved in anterograde transmission. Clathrin is involved in receptor-mediated endocytosis. Vesicles on which of the following pathways would be expected to have COP1 proteins on its surface? So A. RER to cis Golgi, B. RER to trans Golgi, C. cis Golgi to RER, or D. nucleus to RER. Now we know that it must be between the cis Golgi and the rough endoplasmic reticulum. It can't be trans because trans is at the very end, and it can't be the nucleus because the pathway is ER, Golgi, and then membrane, plasma membrane. So it's either cis Golgi to RER or the other way around. Now it's asking for COP1, and it tells us in the question that COP1 is responsible for retrograde transmission. And retrograde is essentially the opposite way. It's going backwards, and it's going from the cis Golgi to the RER, which is the correct answer. Now last question. The steroid hormones produced by the smooth endoplasmic reticulum would be stored in which of the following organelles? A. Secretory vesicles, so the hormones could be released when needed. B. Peroxisomes, so the hormones could aid in lipid breakdown. C. Lysosomes, so the hormones could aid in the destruction of excess secretory products. Or D. Steroid hormones are not stored as they are able to diffuse through lipid bilayers. And so steroid hormones are made from cholesterol as lipid derivatives. And since they're made by cholesterol, they cannot be stored by lipid bilayers. And since secretory vesicles, peroxisomes, and lysosomes are membrane bound, they cannot store steroids. Now if the question had asked about peptide hormones, then choice A would be true, because peptide hormones are stored in secretory vesicles. But for steroid hormones, choice D is the only one that is correct. That's all for today. Next time, we will dive into the respiratory system.